Hi class, I wanted to take a few minutes to go over the different cell walls. So up on the board I have the gram-positive bacteria basic structure. So this thick area is the peptidoglycan, which is your alternating subunits of NAGs and NAMs. That's all that peptidoglycan is. Peptidoglycan is also the classifying feature of bacteria. So all bacteria have to have peptidoglycan in order to be classified as a bacteria. So gram-positive has peptidoglycan and it has a cytoplasmic membrane. That's it. So I want to talk about it in conjunction with the gram stain. If you look at the overall image of this, it is very porous. They're loosely bound together, and your cytoplasmic membrane is selectively permeable. So that you have to keep that in mind as well. Right here, this would be inside the bacteria. So if we think about the gram stain you added the crystal violet, okay? What happened is the crystal violet came through here, through your peptidoglycan, through the cytoplasmic membrane, and then it comes into the bacteria. All right, so you fled it on through. Also, I forgot to mention that within here, you have built in what are called teichoic acids. Those are used for strength, okay? These peptidoglycan layers on a gram-positive can be up to 30 layers thick, so very, very big as far as structure goes. So they need support beams in place. Just think about your skyscraper and how they need support beams, same exact thing here. All right, so we added our crystal violet. It comes on down. Then what happens is you add the iodine. The iodine is the mordant. So once the iodine comes on, what happens is it's basically sealing everything in place. All right, so for a gram-positive, you've now sealed it in place. Your next two steps are to rinse it, okay, with the 95% ethyl alcohol. That will remove any additional stain that just happens to be left over. All right, so that's going to come off. And then the last step is your safranin that you add. And again, once you do your wash step, that's just going to wash right off. So you have 4 reagents with regards to your gram stain. You have crystal violet, grams iodine, which if it's a gram-positive will set the crystal violet, the ethyl alcohol, which is used for rinsing, and then the safranin. So that's your gram-positive cell wall. Let's come over here and let's take a look at our negative cell wall. You're going to see structurally it is very different. All right, so here we have our gram-negative. On the outside, you have a lipopolysaccharide, all right? And then your peptidoglycan is here. It's a very thin layer, and it's sandwiched between 2 periplasmic layers. The periplasm is gel, so it's basically surrounded in gel. So you have a fatty sugar on the outside that's adhered to a partial cytoplasmic membrane, because otherwise they would have nothing to hold on to. Then you have a gel layer. The peptidoglycan is your most porous thing here, and that's why it's sandwiched all the way in. Gram-negative is a naturally stronger bacteria than a gram-positive. Then you have your cytoplasmic membrane. I just didn't draw it the whole way out like I did on the gram-positive for you. Okay, so you have a lipopolysaccharide, a periplasmic layer, a thin peptidoglycan, periplasmic layer, and then cytoplasmic membrane. As far as the gram stain goes, when you add in the crystal violet, all right, this is a lipopolysaccharide. It's a fatty sugar. Can't go anywhere, so you'll just sort of have it all around. When you add in the iodine, same thing. Can't get through anything. That's a fatty sugar. So that's why the decolorizer here is so important. Even though you have rinse steps after your crystal violet and after your iodine, you're getting rid of some of it. You're not getting rid of all of it. You come in with the iodine and you get rid of all of this. Also, what it's doing is it's helping to break down that lipopolysaccharide. Now, underneath it, don't forget, you still have this periplasmic space. So what you're doing is you're hoping to get some safranin somewhere to adhere to that. Now, gram negatives can stain from the lightest of pinks to a sort of a bolder pink. And that's due to how much it's able to attach. If you have a gram negative that stains very boldly pink, then the safranin was able to go ahead and it was able to adhere much better. But in my lab, I have some that are very, very light, some that are very, very dark. It all depends on the organism itself. Okay, so one of the things that I saw on a lot of the answers for the lab was that you wipe out the lipopolysaccharide in the periplasm and you're staining the peptidoglycan, and that's not necessarily true. The alcohol is definitely going to break aspects of it down, but you're not wiping them out. You're basically creating pores so that the safranin can get through and then it can adhere. So I hope that that clarifies it for you. I wanted to go over the basic structures for you because it's very, very important. This is going to be a topic that we're going to be building upon from now all the way until the end of the course. Please let me know if you have any questions.